Hello students, welcome to the biology class. Today we are going to start with our new chapter from the same unit, plant physiology, plant growth and development, in which we will discuss only about the plant growth regulators because the rest of the topics of this chapter is not in the curriculum of Cbse academic session 2020-21. So we are going to discuss plant growth regulators, which we can also being known as plant hormones. Plant growth regulators control growth, development and movement. Growth regulators are necessary for growth development but do not control many aspects of plant growth and development. So it is better named as growth regulators. The effect on the plant physiology is dependent on the amount of hormone present and tissue sensitivity to plant growth regulators. Substances produced in small quantities by plant and then transported elsewhere for the use. So that's known as the chemical hormones. They have capacity to stimulate and or inhibit the physiological processes. So they may be known as the promoters or inhibitors. At least 5 major plant hormones or the plant growth regulators are: number 1 is auxins, cytokinins, giberellins, ethylene and hibiscus acid. So the general functions of your plant hormones: auxins, that is cell elongation; giberellins, that is cell elongation plus cell division, is equivalent to the growth of the plant; cytokinins, that is a cell division plus inhibits the senescence; hibiscus acid, that is the abscission of the leaves and the fruits; and dormancy induction of buds and seeds. Ethylene promotes senescence, epinesty and fruit ripening. So the first we are going to discuss the hormone, that is the auxin. It is growth promoter. Auxin increases the plasticity of the plant cell wall and is involved in the stem elongation. So this is the apical tip where the auxin is present. So the occurrence of the auxin is in the apical portions. It may be the stem axis or it may be the root axis. So the Wendt in 1926 determined auxin enhanced cell elongation. Discovered as a substance associated with phototrophic response, means when the plant is responding towards the light, occur in very low concentration, isolated from human urine, in coleoptile, means that is the tip of the monocot stem, differential response depending upon the dose. So IAA, that is indole acetic acid, then tryptophan and dichlorophenoxyacetic acid 2,4-D. These are known as the synthetic auxins. IAA 2,4-D, which is act as the weeds. So the auxin promotes activity of vascular cambium and vascular tissues. It plays a very important role in fruit development, cell elongation, acid growth hypothesis. Auxin works by causing responsive cell to actively transport hydrogen ions from the cytoplasm into the cell wall space. So synthetic auxin like IAA24D widely used in agriculture and horticultures, prevent leaf abscission, prevent fruit drop, promote flowering and fruiting, and control weeds. So 2,4-D is known as your synthetic weedicide. Additional responses to the auxin, that is abscission, that is a loss of leaves, flower initiation, sex determination, fruit development, and apical dominance. The next is your Giberellin, discovered in association with and in 1930s, that is Bacchini, or the foolish seedling seeds of the rice, is due to the fungi that is known as Giberella fusicoria. So in 1930s, Kurosawa and the colleagues were studying plants suffering from Bacchini, or the foolish seedling disease in rice, and the disease caused by the fungus called as Giberella fusicoria, which was stimulating cell elongation and division. Compounds secreted by the fungus could cause the Bacchini disease in uninfected plants. So he named this compound, that is Giberellin, which helps in the stem elongation. So Giberellin are named after the fungus, that is Giberella fusicoria, which causes rice plant to grow abnormally tall, means that looks like a foolishness. Synthesized in the apical portions of stems and the root, means in the apsis. Important effects on the stem elongation. In some cases, hastens the seed germination. So effects of Giberellins: cell elongation. Giberellin induces cellular division and cellular elongation, whereas auxin induces cellular elongation alone. Ga stimulated elongation does not involve the cell wall acidification, characteristic of auxin induced elongation. Breaking of dormancy in the buds and the seeds. Seed germination, especially in the cereal grasses like barley, not necessarily as critical in the dicot seeds. Promotion of the flowering. Transport is non-polar, bidirectional, producing the general responses. So these are the important effects, or you can say the functions of the Giberellin. The main function of the auxin is cell elongation, whereas the main function of the Giberellin is cell elongation, cell division, and they also helps in the tallness of the plant, that is your stem elongation. So with this, we finish our two important plant growth regulators. In our next session, we will discuss about the cytokinins, we discuss about the ethylene and hibiscus acid. Till then, stay home, stay happy, and have a nice day. Thank you.